Welcome back. Thank you very much for coming back after that short break. Hopefully you've now got a cup of coffee or tea or a bit of breakfast, whatever you need for the next few hours. So today we've got a fantastic panel ahead of us for the next 45 minutes. The session is on, we're going to address the theme of the cell and gene therapy market of the future, monopoly or polyopoly, by breaking it down into 3 pillars for discussion. Those are on the first pillar being the market overview and where it could be heading, a focus on the production, how safety implications and how recent events could impact the future. And we'll aim to cover how these trigger the commercialization landscape within the pharma and biotech space. So the first thing, obviously, it's obviously not just me talking about this. We've got fantastic speakers here today. They are here to answer questions that you may have. We had some questions in the previous session that I couldn't get to. I'm encouraging you all to post your questions in the chat or in the question box on the right-hand side of your screen, and I will get to them as we go through. So I'd like to ask the speakers to introduce themselves for this session. So firstly, I'm delighted to be joined by David Setboun from Brainstorm Therapeutics. David, just an explanation of your role and what you do at Brainstorm.
Yeah, thanks for the opportunity. So I'm the Executive VP and Chief Operating Officer of Brainstorm Cell Therapeutics. This is a publicly traded company. We specialize in neurodegenerative diseases, and we have been developing for the last 15 years autologous cell therapy. We've closed the phase 3 clinical trial. We had a recent phase 2 clinical trial. So we are aiming at finding treatment and improve life of a patient that suffer from ALS, which is a deadly disease and for which there is no cure and there is really little hope. And we're working as well in progressive MS. In addition to the autologous cell therapy side, in the earlier phase, we're developing exosomes, so more for respiratory indications. So it's a great pleasure to be here, and thank you all.
Thank you. Thanks very much, David. And then Oscar Segurado, who is the Chief Medical Officer at ASC Therapeutics. Oscar.
Thank you, Izzy, and thank you, your team at Reuters Pharma. I'm Oscar Segurado, Chief Medical Officer of ASC Therapeutics. Our company has been developing gene therapy and gene editing programs for hemophilia A, and we have a second generation factor VIII gene that we presented to the FDA, and we achieved IND clearance to initiate our clinical trials. In addition, we are also using CRISPR therapeutic approaches to develop factor VIII in vivo treatment, also of hemophilia A. And additionally, we have some work on stromal cells. They're called the Segurado stromal cells for the treatment of graft versus host disease. We recently also reached a partnership with UMass and UPenn on maple syrup urinary disease. This is an enzymatic disorder where there are proteins that cannot be disrupted, and they require a replacement. So all this work has been going on in our company for a number of years, and we are again progressing very much into clinical trials right now.
Fantastic, very exciting. And then last but definitely not least, Matthew Hewitt, who is the Director of Scientific Solutions, Cell and Gene Therapy at Charles River.
That's right. Thank you, Izzy, and thank you to Reuters and the organizers. So, as you said, I'm Senior Director of Scientific Solutions for Cell and Gene Therapy at Charles River. I take a leading role in setting a lot of the strategy that Charles River is taking in the cell and gene therapy realm, as well as running one of our CDMO sites, the Center for Process and Analytical Excellence. Charles River is classically known for providing animal models as well as safety testing. for various programs, but recently we have done a number of acquisitions which have brought us some significant capabilities in cell and gene therapy. The end result is that we have an umbrella, more or less, that allows you to take something from concept to commercialization within a single ecosystem, being Charles River. By training, I'm a molecular immunologist. I've done, AAV gene therapy at Penn with Jim Wilson, was there for a bit, as well as an industry running cell therapy programs, and most recently at Lonza.
Fantastic. Thank you very much for joining us, Matt. A reminder again to the attendees that please do put questions as they come up. I will be tackling them throughout. So first of all, let's have a look at the market overview. So the title. So let's first look at this in terms of monopoly or polyopoly in the future. Will this be an industry of the few or the many? And on a preparation call a couple of weeks ago, we came at this in a couple of different ways. And I wondered whether the panel could kind of kickstart by explaining how the notion of monopoly or polyopoly could be applied to. There's three things here that you need to address, so maybe get your notepad out. So firstly, the C and GT market share by company, for example, big pharma share and biotech share. Secondly, different C and GT modalities, such as allogeneic therapies over autologous. And then the third one would be different therapeutic areas. So I'm going to start with you, David.
Yeah, I mean, great question. I think on the debate and the other panelists are going to jump in, I'm sure, but we believe it's a multiplayer and it's going to remain like that. We had in our discussion the feeling that with the more treatment that getting approved and the explosion of the CAR T cell, we saw much more pharma companies getting into these modalities, and we're seeing being pretty much spread across. We see different approaches depending on the size of the treatment that's going to be used or the production needed. They're getting through a CDMO or even planning of doing their own production. So we see it as being a work where many companies are gonna flourish. They I guess someone like Matt would give it even more insight on the manufacturing, where I believe there is not many CDMO that have full end-to-end solution either. So there might be at one point someone that really came out and established himself as a large leader. But so far, we more see the multiple companies playing in this space.
Okay, very good. So Matt, do you want to comment then?
Sure. Yeah, no, thank you. I again believe it's going to be a polyopathy. I don't believe that there'll be a single or a few companies that will dominate. I think there will be much like other spaces that we see where there's a lot of innovation continuing to come out of academia, as well as small, agile biotechs. That being said, I do think it goes beyond a little bit the therapeutic developer side, as David was alluding to, in that, if we look at a lot of the field, I would say around 3/4 or so, according to most of the surveys, are allied in some form with a CRO or a CDMO. I think that's likely to continue for a number of reasons. The primary one being that the demand curve for what we hope are curative therapies is far different than maintenance therapies. And then so how you design and build a manufacturing facility is going to be quite different in that case. And since you're going to be reliant on utilization as your primary driver, every company builds their own manufacturing facilities to control their manufacturing destiny, which I fully understand. I think there'll be two problems. One is that they'll be inefficiently used, and secondly, the manpower needs or the people power needs are going to be massive. And we're at a point in the industry right now where there's a massive talent shortage anyway. And, a lot of specialized training goes into GMP manufacturing. Allo versus auto, I think they'll coexist. It's a very easy answer for me. I don't think that one will necessarily dominate the other. I think some of the results we're seeing in autologous, say with cilta-cel for multiple myeloma are, frankly amazing. And I don't know if we're going to get too much better than that. And then at the same time, there's going to be indications where allo is going to work better than auto. In terms of modalities, obviously right now T cells dominate. In CDMO and CRO, we are seeing other modalities come out, NKs, monocytes, myeloid cells, MSCs, of course, and iPS-derived cells. But at the moment, it seems like the field is still dominated with T cells.
Fantastic. Thanks very much. That's great to see the kind of the different perspective that you have on your side of the fence. And then Oscar?
So I see a coexistence of polyopoly and monopolies. And that's going to be related to the different stages of development. In the very, very early stage, we have a number of academic centers that are going to have proof of concept studies. They will be able to just do some studies in the lab. And there are going to be companies, biotechs like us, that are going to acquire these or license this know-how and start bringing it to a stage where it can be presented to the agency, doing the so-called IND enabling studies. And from there, if we are successful, if we are able to conduct phase 1, 2, and even pivotal phase 3 studies, then we're going to have a number of pharmaceutical companies that are going to start looking for partnerships. So the way I see this happening is that in these early stages, there is going to be a polyopoly. We know that there are over 5,000 genetic diseases and over 100,000 genes that have been associated with either these diseases or to very common others like diabetes or heart disease. So this is going to be a very, very broad type of companies and academic interactions once they have solidified and still being very broad. I think there is going to be a kind of monopoly with a number of companies, with pharmaceutical companies being able to afford the risks associated with reimbursement of gene therapies, especially, and also cell therapies. So there are going to be these big pharma that are going to really drive a number of products going on. There is also a second aspect that is going to be relevant here, and this is the distinction between gene therapies and cell therapies. In gene therapies is where I see this coexistence and this amount of different type of companies working together at different stages. But in cell therapy, it's going to be even more concentrated. We're seeing that with CAR Ts, with everything related to genetically engineered T cells, we have only a few companies, especially in Novartis and BMS and Gilead, that are really leading the pack. That's not going to change easily. That's going to stay for a while.
Fantastic. So some excellent points there. And we will move on to another question, but still in this kind of like market overview discussion. So we're currently in a very advanced commercial stage for autologous cell therapies, but with a big push for allogeneic gene therapies, gene editing, and others. Can the panel provide an overview of the scientific advancements and really kind of an investor perspective for the different modalities? We'll start with Matt.
Sure. I mean, I think that focusing firstly on auto versus allo, I mean, allo is attractive because the manufacturing becomes a bit easier, and easier is always a relative term. But you go from having to manufacture a single doses for every patient to now having a central donor that can treat multiple patients. So therefore, you can amortize the cost of that manufacturing over a larger number of doses. Of course, there are a couple different ways to do this. If we're talking about donor-derived starting material, then having your donor profiles mapped out quite well is extremely important. We do have that, for instance, at Charles River and Hemocare that we have recently acquired. And I can tell you that the donor profiles, are increasingly becoming more and more complex as people zero in on what they're looking for. That has been advancing well. I think the regulatory is still playing catch up a little bit there, especially if we're talking about getting away from donor-derived material more on the iPS-derived side. Looking at gene editing, I think that gene editing is the future. I think it's the way that we're going to be able to leverage this field to its maximum potential. I do think there are some issues in that a lot of the IP within gene editing is concentrated into just a few entities. And I think that it's going to take a little bit of a shift in the way that we're thinking and how those licenses are given out, because it's not feasible for a small startup to pay large upfront sums for a single program to get going. And I do think that is a barrier. I do think that there is an opportunity to use this term that a lot of people are using right now, which is to democratize gene editing. The reason why most people are still in lenti, in some cases gamma retro, is because they're largely royalty free. And so if we're going to get into gene editing and we're going to see it really spread throughout the field, I think the advances on the science side have been considerable. I did work with, the original Cas9 many lifetimes ago, and it's improved considerably. But if we're going to continue to chase, these large licensing deals, which is I certainly understand the thinking there, and it's reasonable for people to do that, we're going to have a little bit of trouble in terms of spreading that technology throughout the field.
Right, good points. David, you were nodding.
Yeah,
I think
I can jump in a little bit. I think from an investor standpoint, and coming back to Matt was saying at the beginning, I don't think either that would be one modality that will win it all. I think there are really disease specific. So you would need autologous treatment. You need to personalize some of the treatment. There's a lot of trend into personalized medicine and oncology as well as the benefit of having an easier production if you go for Allogeneic. So I think both of them are going to coexist, and it will really depend in terms of the interest or the type of disease, what exactly you're trying to achieve. Both of them have as well some limitation, one on either on the production side or on the type of the donor that you're looking at. So I have a more like positive view that those technologies are going to be more used, and the more we have seen a big splash with the CAR T cell, but I think it's very likely that we will see other treatment being approved in other disease area in the cell therapy, and that will open the field dramatically. I think it's a really a new era for many disease and many treatment, like large molecule were 20 years ago. So I think this is going to be thriving and accelerating. I agree that the gene editing, it's where many investors are going right now, and it's there is too large IP concentration and company that has been very, very high valuation behind those, and that provide a lot of possibilities for many cell therapies, and it is going to continue. So in other words, it really depends where the investor want to be and how close to a treatment effect, or how close to be to the clinic he wants to be. But I strongly believe that we're going to see more and more cell therapy approved in the next 5 to 7 years.
Fantastic. Oscar, what are your thoughts?
So I'm hearing a common theme on these two topics. The one, allogeneic versus autologous, and also the issue that we have with IP on gene editing and base editing and all these approaches that obviously are very relevant for the field. Starting with autologous.
Oh, we lost Oscar.
And allogeneic is a few years. Can you hear me now? Can you hear me better now?
Yes, we can hear you, Oscar. Sorry, we just lost you for a minute then.
Okay. So yeah, I was going to touch on two topics. The first, autologous versus allogeneic. We have made a lot of progress in autologous therapies. It started everything at a number of universities in Seattle, in UK, in several places. And everyone is now very, very comfortable with the results that have been already especially seen in a number of hematologic disorders. So we practically manipulated the T cells. We were able to get them being very well targeted to certain hemato-oncology problems. And from there, now we want to ensure that we are able to do that, not just patient by patient, that is the way it currently works. In lymphocytes, the work that has been mostly conducted at the NIH. and a number of companies. Then we're trying now to get into allogeneic. That's a big leap to be there, and there are a number of companies like Allogene and others that are trying to work there. Another approach is to be able to use these stromal cells, like we are doing with the Segurado stromal cells, that can be used as a kind of an immunoregulatory type of cellular approach to certain cells. So there are an extraordinary technology approach by several companies, and I think that's relatively a free field. So in one sense, this is an all you can eat. So everything that you can do from a technological or scientific perspective, you can practically move it on through the development programs and bring it to the market. On the other hand, when we start talking about the situation with gene editing, with CRISPR-Cas9, and all these type of patents that are required for you to be able to commercialize, I agree with David and Matt that we have a problem here. That there is, and we know that because we have in-licensed CRISPR, and it has, in our case, it has been regionally focused. And these are very, very expensive type of deals that you need to reach with a number of companies that really control that space. We know that that's something that I agree that we should be able to alter, and this is really slowing down probably the development and future commercialization and help for many patients, because we all know that in vivo and especially ex vivo gene editing can have a tremendous impact on many diseases where you don't really need to completely transfer an entire gene.
Yeah, I might add maybe two things. One is on the cell therapy side, and Oscar, you I think brought this point. I mean, the data that we have in cell therapy as an example in oncology for hematologic indications is very, I think, pretty solid. And I think we know how to deal more or less in those hematologic indications. What we're seeing now, obviously, is everybody's moving to solid tumors, which makes perfect sense because year over year, hematologic indications account for about 6% of tumors. So we're really just scratching the surface. What we have seen in some of the clinical trials is certainly some data that isn't consistent, and we've also seen some more severe toxicities that we haven't really been able to control yet. So I think that is something that is on the horizon and investors are looking at. On the gene therapy side, obviously recently we've had some severe AEs in a couple trials, as well as a recent meeting from AdCom at FDA to talk about potential maximal doses, as well as presentation of foreign protein to patients. And I think these are all areas that as investors, they're going to start looking at them more carefully, because obviously we want programs to move quickly through development, but we want them to move safely through development as well.
Absolutely. We're going to switch. Well, we'll talk about safety. There's a few questions down, but I just want to, there's a kind of a sub question. Obviously an important stakeholder here is academic institutions. Indeed, we've probably got a few attendants here. So I'd just be interested to look at kind of like where we are currently in terms of our relationship with academic institutions as a stakeholder, and what the possible future may hold, and whether we see that changing. Who wants to pick that one?
I can
David?
start. Yeah, I can start.
David.
There is, a lot in the US, and we see it outside of the US as well, but there is a lot of opportunities to partner with academic centers in the cell and gene. We see more and more large centers. They are moving to be much more, let's say, able not only to have early program, but to get into early manufacturing as well, and an ability to produce. I'm thinking about EVs specifically. So you can see a maturity of those academic centers, and I think they are fantastic opportunities, because not only they're bringing early innovation, and they have an ability to get a regular GMP production, and very often they belong to centers. So you can have your early clinical or your first phase 1B clinical trial to get started in the same center. So we do see that move where maybe some years ago they were getting only to the academic discovery, very early preclinical. We see many of those large academic centers moving into later preclinical work up to IND, or even taking risk themselves of moving towards their own phase 1B and looking at partnering with pharma or biotech a little bit later. So that's a trend that I'm seeing. I can say that I see a little bit more being done in the US than in Europe, for example, some emerging other in the cell therapy in China, but I see more large centers. So that I think it's a great trend in terms of partnering for both biotech and pharma companies. But the bigger they become, the bigger the deal is going to be, and they're trying to attract much larger deals as well than early VCs. So yeah, it's partnering for companies, partnering with a lot of VCs that be very aggressive in that space as well. So yeah, it's a positive, and the dynamic is there and thriving, I believe.
Fantastic. Oscar?
Yeah, I think we're touching something that is absolutely relevant to the collaboration between academia and biotechs, and then later on with big pharma. And this is the aspect related to safety, which, as Matt just mentioned a moment ago, that was the focus of a 2-day FDA meeting talking about AAV aspects related to safety of gene therapies. And what is absolutely clear right now is that you can see a relatively safe result in animal models. And the way we use translational medicine, translational science to take that into clinical trials is related to luck in some way, but also to the dose that we are using on these therapies. So in terms of gene therapy, is very different with the so-called vector genomes per kilogram that you use, and that's why you need companies that have a lot of clinical experience on being able to move these studies through clinical trials and ensure that you reduce, by the way, this vector genome per kilogram as much as possible. So that was one of the outcomes, or one of the highlights of that meeting. So that shows how important it is for academia to ensure that at some point they hand over all these results to biotechs that have a very strong clinical know-how to be able to understand that. And in terms of safety, in terms of a number of deaths, even that have been recently reported by a number of companies, I think that shouldn't stop all the developments that we have been achieving over the past almost 1 or 2 decades. And that's also because there's an aspect absolutely relevant here. And this is the type of disease, the target that we are using with the gene therapy. It's not only the fact that we're using AAVs, the fact that they potentially can integrate into the liver cell, that they can also have an immunogenicity that could have certain deleterious effects when treating patients. It's also ensuring that we see different compartments on different diseases and therapeutic areas.
So, I mean, I will touch on this, and we might as well go to the safety right now. Matt, I'll go to you first for this question, if that's all right. So it was really, the question we had on that was that there have been, obviously there's the FDA meeting we talked about. We've also had the kind of 4th death in gene therapy in the last couple of weeks as well. And it's brought up, raised some concerns around safety. How might changes in risk perception around cell and gene therapies impact the future of the industry? Matt, I'll go to you first.
Right. I mean, I think you have to bifurcate this a bit into gene therapy versus cell therapy, because the risks considerations are going to be far different. In gene therapy, and this is a little bit coming back to the prior point around academic centers, a lot of this is done in academic settings in the beginning. And then, in gene therapy, you're going to be moving into a fairly large batch of GMP material to go into your early phase trials just because of the amount that's needed. The models that we use, as Oscar was alluding to, are not perfect. Certainly most of the time, because you're injecting an animal, whether it's a mouse or an AHP, with a massive amount of AAV with a human transgene, and then you're trying to look at transduction efficiency as well as immunogenicity of essentially a foreign gene into a different animal. On the gene therapy side, I think we're dealing with a lot of rare and ultra rare diseases where there are few, if any, other treatment options. And so I think the risk profile there is going to be far different. They're probably likely to be a bit more lenient in trying to get patients, especially children, options. But obviously at the same time, when there are deaths, we need to pause and do some fact finding there to understand what went wrong. On the cell therapy side, I think that they're, much like in oncology in generally we start with the sickest patients and then move our way forward from there. And I don't know if that's going to necessarily change that much in the future. I think that will continue to be what happens. There are other options, obviously, with chemotherapy, radiation, and other immunotherapies that are quite good. And cell therapy right now is a little bit further back in the treatment paradigm. However, I would say that given the clinical results we are seeing in these patients that have been heavily pretreated many times, that bodes well for what happens when we likely move it forward in by the lines of therapy, because many times we're dealing with cells that are in very bad shape from these patients, if we're talking about autologous, and we're still seeing considerable results. And again, maybe wrapping that around the previous question, the academic centers are absolutely critical for this. They've been doing this as well as NCI for a couple of decades now in some cases, and they have the experience, they have the people. They're great for early phase clinical manufacturing because it's closer to the patient. So it's going to be definitely a symbiotic relationship here between therapeutic developers and the academic centers to de-risk these programs.
And I'm just gonna jump and add on the safety side. So I think it was said by Matt. I think it's very, very important, first of all, to differentiate the technology. So I haven't seen signal in cell therapy that are alarming. And to what we've been seeing recently, both in terms of efficacy and safety, we see more and more program with some strong safety patterns. So let's say this is on the cell therapy side. On the gene therapy, I would agree with what has been said. So if you look back years ago, there have been some issues. I think we're way past that. There are people treating with gene therapy today. Treatment are being commercialized. The benefit risk ratio is very strong. It's very, very sad what happened to the patient, but I don't think the last 30 years of development and research are going to stop, and it's more of a better understanding and to continue to move forward. So there is, I guess, strong potential for gene therapy moving forward. Obviously, we need to stop and pause and understand, but that will not change this drive. And again, I think on the cell therapy side, you see more and more, and to the extension to additional indication, I think it is going to continue. And I can see that patient and the investor community become more and more at ease with the safety of cell therapy overall versus what they had in the past. So the cell therapy are going to be maybe more challenged by the ability to manufacture those treatments at a larger scale. But if the manufacturing is so tight that you will release treatment that are safe. So the safety would become a second, let's say, issue for the cell therapy. And then obviously we're going to mix both. So then that's get another level of complexity.
Yeah. It segues nicely to the next section we've got, which is production. But before we go on that, I do have a question from the audience from David Hines. Thank you, David, for your question. It's he says, stemming from that, do you see the necessity for academia to adhere to stricter regulation, structured processes, e.g., or GMT, etc.? Sorry, GMP, etc.
I mean, I think that most of the academic centers already do adhere to GMP. In the case of cell therapy specifically, there aren't many that are doing gene therapy manufacturing on site just because of the scale that's needed. The one exception to that might be Australia, of course, because they have a bit of a laxer requirement on your phase 1 material and what level it needs to be. I think that the other consideration is in cell therapy specifically because neither the plasmid nor the vector are the final product as it is in gene therapy. There are some leniency. There is some leniency given in how that material is produced. It still goes through much of the same test. It's just it's not full GMP, and so that's when you see plasmid production from that run at GMPS, or in the case of Vigene here at CRL, we have a high quality or GMP ready, and then that goes for plasmids as well as vectors.
Excellent.
So I like to add that, that after 10 years, mostly focusing on cell and gene therapy, I can tell you that everything related to GMP manufacturing is something that even for someone like me leading the clinical programs is absolutely relevant. So, and the same applies also for academic institutions. They also need to be aware of everything that is required by the agency, and also that when developing any type of product, when handing it over to a biotech or to a pharmaceutical organization, that they need to ensure that they understand that they're very good examples. I worked at some point of interaction with Children's Hospital that already was doing that. The same applies to us. Right now, we are also working in Ukraine and UMass, and we know how important it is to do that. Now, let me also say something. When I started working in this field, working with biologics, with the monoclonal antibodies, I was working with the Humira team at AbbVie. We thought that was a kind of fighter jet. It was extraordinarily complex. Now, what we are doing today, both in cell therapy and gene therapy, is like building a space station. And when you do that, you not only build one, you have to ensure that you are also able to replicate that. So that's tremendously important. Manufacturing and being able to produce your drug is something that as a chief medical officer, usually I don't think much about that, or I didn't. Now is the contrary. Now we have to incorporate that totally in the way we produce and also how we ship.
Absolutely, and that does bring up very
how we put that drug into the.
Absolutely, that's good. Brings us very nicely on to the next question. So on the preparation call, we identified cell therapy growth and a focus on manufacturing capability as a huge opportunity in this space. And David, you said that manufacturing is now a prerequisite for any commercial thinking in cell therapy. How will the industry need to adapt its manufacturing strategies? And again, how might big pharma and biotech be tackling this differently? So we'll go to you first, David.
Yeah, when you think about it, I mean, I've done my career in commercial and large pharmas. We never thought about manufacturing. It was more, okay, how many patients are we going to do? And then we can say, okay, we're going to figure this out. And someone is going to produce these small molecules. It's going to be pretty easy. So now it's a little bit the other way around. It's what I was alluding to is when before you were needed to have everything ready for your commercialization of the treatment, you were thinking about your medical resources, pharmacovigilance, and the manufacturing was a given, and even the patient journey was a given. So you didn't have the need to really follow the patient, get the cells from the patient, and having to plan all of these very intense logistics. So to your point, everybody thinking along those lines now and making sure that you have your manufacturing capability ready. We see many CDMOs stepping up massively those days to be able to enter into this commercialization phase. And we see as well more initiative of companies trying to produce themselves the treatment. So to your point, that's a fantastic opportunity. I like what Oscar was saying. I think we are like maybe large molecule 15, 20 years ago. So I can see the move, I can see the structuration, the consolidation of manufacturing potential, the start of the automation as well. You can see a lot of things going in the right direction. It's going to take some time, but I think it will become more direct. But anybody that want to commercialize need to plan very well. His manufacturing is almost prerequisite to any thinking of commercialization now.
So Matt, to you, see what wider impact will this have on CDMOs across the cell and gene therapy modalities? And do you see more and more pharma and biotech taking manufacturing in-house in the future? I mean, obviously, we've seen that with GSK and Novartis and others.
I think it's going to be more and more rare as we see more and more therapies come to market, again because of the staffing needs. The technologies are certainly, coming. They're coming up. I mean, it's not expected that we would come into this field with everything we would need right away. So, now the technologies are coming up. At Lonza, for instance, I led development of the cocoon, and we did a lot of really great stuff with the cocoon. I think it's going to be a good option. I'm not saying it's the solution. It's going to be one solution. It's going to be one tool in the toolbox. But that's an example of what we're going to move to, is these closed, automated, and I think most importantly, scalable manufacturing solutions. It's a little bit different, again, if you bifurcate this into gene and cell therapy. So for gene therapy, we can batch this, and geography doesn't matter as much. For cell therapy, it will, because of the logistics that are required, and that's something that we're still working out. And it's why we need this technology in these closed, automated, scalable systems to couple with the logistics, so that we can make this much more efficient of a process. So when you reach step X, it notifies a stakeholder that now it's time to bring the patient in and begin preconditioning. There's a lot of stuff we need to do around releasing a product to hopefully speed that up. And then I think the other one is looking at the centralized versus decentralized manufacturing paradigm. Having one location in North America, one location in Europe, maybe one location in China, and one location in the rest of APAC is going to be doable. But again, I think the logistics are still going to be very, very long. There was a recent poster at EHA from Shiba showing that when they were, bringing in a cell therapy, they had about a 20% or 28% out of spec rate. And a lot of times what would happen is it would take so long to get that therapy in that a lot of patients would have disease progression and then couldn't benefit from the therapy. So, I do think we're going to have to put the manufacturing locations closer to the patients. I don't think it's in the clinical center, but certainly near it. And then what we will then have to deal with, again, coming back to the logistics, is supply logistics to those different decentralized sites. By decentralizing, you can also potentially stratify your workforce if you do a hub and spoke model, where the spokes are producing the therapies and the hubs act as the brains, they're actually releasing them. And that might be a way to alleviate some of the problems with people, which is always a consideration.
Yeah, Oscar.
I just want to add that there is a very important aspect when we talk about biotechs working with contract manufacturers like Charles River. We started working with Vigene, that was recently acquired by Charles River 2 years ago. And because we have some expertise through our R&D enabling studies on analytical methods and several aspects, it's extraordinarily important to just not use I'm using that word, a contract manufacturer to do all the work. You have to work very closely with them, and also with and ensuring that you get the entire package ready for GMP production.
Fantastic. So we've got about a minute left. I'm just going to go around, and if you can do very brief, it's a very big question. So kind of what do you see as the kind of the biggest opportunity in the business that you're in for making an impact and getting these treatments to patients? I'll start with you, Oscar.
So I just want to say that this is the most exciting area I've ever been. I'm thrilled about the opportunity to work on an area where we understand we not only know the genes, but we also understand their function, and we're able to genetically engineer that. That's absolutely amazing. So it's something that is going to change the way we see medicine. We're moving into medicine 2.0 now.
David?
Yeah, the biggest opportunity is really the ability to target a much broader population. And I think that will come, and thinking that you can apply those high-end technology for larger disease are going to bring solution to many, many patients in the future. And that's what is very exciting.
Absolutely. And Matt, finally?
I mean, I think gene editing and synthetic biology are the two most exciting areas. I do think that an interesting question I'm starting to think about is, where do we cross over from gene editing to genome writing? Because now people are talking about doing many, many edits, and I'm just wondering, when do we shift from editing to actually genome writing? And I think that's going to be an important question soon.
Fantastic. Well, I want to say thank you very much, David, Oscar, and Matt, for your time today, for sharing your insights with us. Indeed, if anyone's got any other questions, do reach out to them on the platform. But also, Charles River have a exhibition booth on the virtual exhibitor hub there, and you are very welcome to contact them there if you want to find out more information about the fantastic work that they're doing. So we're going to move on to the next session right now, which is from Claire Feeney, who is the Global Dermatology Director and Consultant Endocrinologist at Pfizer and Imperial College in the UK. And she's talking about social listening and an uncensored view of real-world patient experiences. Over to you, Claire.